It's an international effort. I'm actually sitting in Colombia, in the west side of Colombia. But we have people from China, we have people from the Netherlands, from different parts of the US. And yeah, so welcome to everybody. Here's the link to our project website. There are multiple people involved, and you will meet several of them as we go along. I just want to highlight that on our website, we have recording of the previous meeting, and as we publish things, as we develop more newsletters, this is the repository that we will be using. I'm going to give a little bit of an introduction, and then we are very, very fortunate to hand over the floor to first to Plasma Treat and to Vital Fluids. And these two companies are well, the cold plasma system I have in my lab was developed and purchased from Plasma Treat, and they have been excellent. Vital Fluids is a company we have we have a lot of a lot of communication with, and are hoping to work with in the future. And the fact that they are on the program represents the spirit of the research we are conducting, which is that we are truly interested in bringing this technology, assist companies to make this commercially viable to growers around the world. That we are doing the research, but the research has a very commercial and an applied angle to it. Then Cornell is going to give an update and a presentation on the survey that we conducted, and then we have some different research topics that we will cover after that. And of course, this research is only possible because of money we have been securing. So we have secured at this point about $2.5 million from federal and state funds, and we are still looking for more funds, and we are definitely hoping that with these meetings we can also identify additional aspects to add to what we're doing and expand the research we're doing with private companies and other universities, and get more students involved, and for them to learn about cold plasma and its use in sustainable crop production. A quick look at who we are in the Nansen lab at UC Davis. Well, we do a lot of robotics and actuation. We do optical sensing. Of course, we're going to talk mainly about cold plasma, but we are also going to talk a little bit about some of these other technologies, because I really think it's important that even though the cold plasma is the main denominator in this presentation and in these projects we are having, we are adding other tools. It's part of an integrated approach. We're definitely technology driven, and we want to combine these different technologies in innovative ways to create new ways of growing sustainable food. Use less pesticides, rely more on natural enemies, sort of overcome the issues we are having with resistance by using lighting and of course cold plasma to manipulate pests in a way that make them more easy to control. So this is part of a much bigger picture, and that's kind of where we are starting. This is the base we're trying to develop, a base that goes beyond cold plasma. And the team at the moment, Miles has since he's still on this call here, is on his way to Florida. But the rest of the team, the key members are listed here. Anil, our technician, our electrician and lab manager, who does all the electronics and programming, and Hale, who takes care of our plants and our cultures, and Patrice, who's a postdoc, and Yu Gong, listed down here, is listed as an undergrad, but will in a couple of months be a master's student. And we have a whole bunch of other students, but Sammy and Sydney are the key ones involved in the research that we will be presenting today. So clearly a team effort. And the team, of course, involved in these proposals that we have submitted and successfully successfully uh, um, we have George at Minnesota, we have Neil, and these are just the PIs because under each of these people there are multiple people, right? So what I've tried on the right hand side is to provide some color codes on what people are involved in, and what you can see from this, of course, is that, and actually I can see that the screen is cutting off the stuff. So anyway, there are many aspects, many dimensions of this. We have, yeah, actually it's completely cutting off the colors. So this is a little bit misleading. I'm sorry. I'm working on a different computer here. So, but the point is that we are very multidisciplinary, and we are still looking for people, especially in the plant pathology, because technology that need to be included. So we're going to be talking today exclusively about plasma activated water. So cold plasma can also be applied directly to seeds and possibly to plants, but we are not going to be talking about that. That will be a topic for we still have research to do in that area. So what we're talking about specifically today is what you're seeing a video of right here, where we insert a jet into water and then magic happens. The list to the right is just some of the variables that completely change in the water that is being treated within seconds and minutes. And it's a very dynamic system. So we are looking at how all of these factors interact, which ones we think are the key drivers in this, and how we can model how water will change, because it also the changes in pH or conductivity, salinity, and sodium nitrite and all these other variables depend largely on the quality of the water that was treated in the first place. So this is just to make a first key home message to people who are not familiar with this technology, that PAW or plasma activated water is not one thing. It's a concept. It's a framework where we can generate many different kinds of PAW depending on the gases that we project into the water and how long we treat and the quality of the water in the first place, and other factors. So what I did as preparation for this meeting, I took 351 samples of PAW that Miles has been running for the last couple of months, and looked at and just did what's called a principal component analysis, which is basically just looking at the association or the similarity, dissimilarity of different variables. And when you put them in a two-dimensional space, so you can see which ones line up close to one another. So the closer they are, the more similar they are responding. So basically what you can see is that the different variables, excluding hydrogen peroxide and ozone, which are variables we don't have as much data on as we have on these other variables, you can basically see that they fall in some families, so that ammonia and ammonium seem to follow each other, and there are some other groups here. What is important about this is that if we can see that there's big, strong association between some of these variables, we may not need to monitor all of them, and we can also see which ones are negatively or positively correlated with each other. For instance, this gives us a clear idea that the oxidative reactive potential is negatively correlated with these nitrogen species that we have over here and the pH. And the resistivity of the water is negatively correlated with salinity and total dissolved solid and conductivity. So we're getting sort of an idea of the dynamics and trying to reduce the dimensionality by doing something like a principal component analysis. And this can really help us in the future if we want to optimize one thing and minimize something else, well, how are we going to treat this? This kind of analysis can help us in that regard. And today, I'm just, because we can't talk about all these variables that we're collecting, I'm just highlighting nitrate, ammonium, and conductivity. And of course, nitrate and ammonium are being highlighted because those are the major forms of nitrogen that plants are taking up. So those are the ones that we are really interested in. So with this dynamic here, what's the potential? What's the potential for crop production? Why is PAW such an interesting technology to look at? Well, one thing that I dug up while I've been down here is this basic question about loss of fertilization during application. So growers, whether they're in greenhouses or in orchards or fields, will apply nitrate and ammonium and maybe some other forms of nitrogen. Well, what happens to that? How much of that is actually lost? And it turns out that those numbers are actually quite significant. It depends on pH and soil conditions and whatnot. But some of the data that I found was that somewhere between 10 and 70% of the nitrate that is being applied is lost to nitrogen as a gas. And the ammonium can also be lost in significant amounts, from somewhere between 5 and 30% to ammonia, which evaporates. So in total, what we're looking at is somewhere between 15 and 30% of the nitrogen that is being applied actually is lost. So this resonates and makes this PAW really, really interesting, because as you will see, we can use PAW to generate nitrate. We can use it as a fertilizer, as a way to, if we have renewable energy available to run our system, we can actually generate PAW. We can generate PAW and therefore nitrogen to our plants in a sustainable way and decentralized way, which is really, really interesting. And the nitrogen budget for plants, of course, is very important to growers, but it's also something that is very dynamic. As we generate PAW, how stable are the nitrate and ammonium concentrations in the PAW? What can we do to stabilize them over time so that we can store, so that we can distribute the PAW in different ways and make it as feasible and adoptable by growers as possible. And again, so what with this dynamic of PAW and with these characteristics of PAW, well, we can definitely create fertilizer. We can also potentially boost the effect of the use of fertilizer. We may even be able to boost rhizobacterial communities in the roots. We can and we are definitely generating data on this. We can make seed germination more uniform. We can speed it up. Seedling vigor can be increased. And also something like transplant shock, which is something that when crops are being transplanted out in fields, this is something growers are used to, but with plants being irrigated with PAW, they may have a different root structure, have a stronger root system, and therefore be able to, as they get parachuted into a field, they may get a foothold much quicker and do better, and therefore overcome that transplant shock more readily. We also think in the pesticide business, and we will definitely show you results on that already here, that there are some direct fungicidal effects. We know that Vital Fluids is very interested in that. We have strong data that Patrice has been collecting and submitting, has published on, and I'll show here in a little bit, that we can boost plant resistance by irrigating plants with PAW. We also have some data on how we can, instead of using water for pesticide formulations, if we use PAW, we may be able to boost the performance of those. So use it as a carrier for pesticides, use it not only for the plants, but actually also in different ways in pest management is definitely something we are very interested in. And then George at University of Minnesota with his expertise and in food science, we're definitely going to look at yields. We are in the process of growing tomatoes and we'll soon be sending them to Minnesota, have them analyzed from field plots. And that's going to be a topic for another meeting in the future. We have a fascinating field study going on where we're collecting drone data and going to collect yield data from transplants. And this is something with Tom's experience and his extension facilities in California, we will be able to make this commercially relevant. We will hopefully demonstrate on a large scale that we are not just BSing here, that this is something that is actually creating different yields, not only in terms of quantity, but also potentially in quality. So these are just some of the key aspects of the potential. And this is why we're so excited about this PAW and why we are so glad you are here today and why we want to share this research with you. And I can talk all day long about this, but I think a photo speaks a million words. Like, so just to give you a little bit of a context here, I mean, this is what happens when you grow tomato plants. Indisputably a difference here. So we can see that. We can also see it when we grow ornamental plants. We can see that marigolds are bigger, they have more flowers. So this is something that really is important. In strawberry, we have seen similar effects, that different kinds of, so you can see there's two PAWs here. So this has to do with the treatment, and this we can maybe even get better results. So is this also going to give us better yields? Well, that's going to be the next step. But just in terms of growth, we can definitely see effects. So with that, we are sort of the key questions that we are honing in on, and what we are hoping to discuss, and this is where I would like to start by opening up a little bit. I think, or we think that what is going to be interesting if you are if you're a strawberry grower, or if you are a marigold grower, or if you're a tomato grower, well, how much PAW is needed, right? What are the characteristics of the PAW that will really benefit your crop? And it's probably going to vary from one crop to another. When should you apply it? How frequently should you apply it? And then how can we store it? Does it need to be produced and immediately injected into your irrigation systems? Can you in the audience think of and Anil, if you don't mind, if you could take notes, or somebody can take notes from the team. Are there other questions that the audience that sort of pop up to the audience here that you think we should be looking at?
Well, this is George here. I would say one of the other questions that we need to be thinking about is, do we have to change that water after we produce the PAW? Can we add other minerals? Can we add other supplement, if I may use that word, to make it more potent for using it for plant growth?
Right. And that maybe adding to that is also, so to me, that falls under the needed characteristics of PAW, but what you're saying is absolutely is a good point. And also, I'm now thinking, what is the importance of the water being used in the first place, right? What is the input? So not only what can we do afterwards, but also do we need, before we subject water to plasma treatment, do we need to do some pre-treatment of the water before we subject it to the jets, right? Mm-hmm. Any other comments from the audience at this point?
Just trying to put my grower hat on as an academic, I guess. A more applied question would eventually be, can I apply it using my commercial fertigation system? So can it go through an irrigation boom and be successful? Can it go through a drip irrigation system and be successful?
That's a great question. I'm sure Vital Fluids, they have some experience, and maybe Paul can talk a little bit about that. Or Paul, is that something you're going to talk about?
Yes, it's actually in my presentation. Well, we have systems running in the US in Morocco, in Spain, and in the Netherlands that are connected to drip irrigation systems. So it's no problem to use plasma activated water for this purpose. So, but it will be in my presentation. Yeah.
Fantastic. Oh, that's great. That's great. Well, anybody, as we go along, feel free to chime in. Feel free to interject. We want this to be as dynamic as possible. So what I was hoping now is if I could let Ryan from Plasma Treat take over at this point. Ryan, is that okay for you?
Absolutely. Thank you so much. I appreciate the very thorough introduction. It definitely saves me some time on explaining the activated water and how awesome it is. It's definitely a passion project. You know, it was all related to you know, my thesis work way back when. So it definitely is nice to see that it's being recognized, you know, for all its other applications as well. But just as, you know, for our company and such, I thank you guys for giving us the brief little bit of time, but also the, you know, the stage to kind of share some of our work recently. And before anybody starts to, you know, go aghast, yes, it is cannabis. I am, you know, saying hello from sunny California as well. So luckily we have access to such things to have a little bit more of a spread. It's nice to have a robust plant to work with. Just as a slightly different approach for application than most of the literature you might find these days for activated water. You know, we actually went at it from a plant cloning propagation standpoint. So kind of working from the clippings from a mother plant and applying, you know, kind of a semi-optimized PAW solution to these plants as they go from being clippings all the way to a root structure being formed and then put into soil and kind of moving on forward from there. But essentially, the reason why we were able to get a collaborator on this particular project was, you know, the shorter that time for that growing portion to actually happen, it's reduced operational costs. This is all indoor, so there's lighting costs, there's water, and just, you know, general maintenance of a building for that extra week or so. So they had some added interest for it on their own right. But we also were able to, from that collaboration, to get somebody that was experienced in monitoring plants. Again, Plasma Treat's more about the production and usage of the atmospheric jets. So when it comes to plants, I have a few plants around my office, but you know, we needed somebody with a little bit more expertise at the end of the day. So we did use a single strain to kind of keep things consistent as well. You can also see that our you know, number of plants, we had a count of around 50 per tray, if you will. So this is a video of a very similar jet to what Christian showed us earlier. They have a stationary jet, as we call it. The one that I'm showing here right now is actually a rotational version of that. So we take the same plume, tilt it off by about 15 degrees off perpendicular, and then rotate it at about 3,000 RPMs. It's the same effluent, but it does its own mixing of that solution of the water that's there. So this is distilled water. We treat this for about 15 minutes. Through some of our initial work, we were able to find that, you know, 15 minutes is about where we start getting diminishing returns on the treatment that we're seeing. But what we wanted to show is, you know, with this added agitation, there's a significant number of, you know, micro bubbles forming in the bulk of the solution. So we're getting a lot of good mixing in there as well. So this isn't, maybe the end production, you know, reasoning or option for in the field, if you will. But it's definitely a good way to get a quick, nitrogen fixation in the volume.
So I have a comment. It's very interesting. You guys also actually dip the tip in the water, right?
So very closely. You see, the distance is actually 4 millimeters. It's basically touching once you start all this agitation and such. But before it gets started, we place it about 4 millimeters from the surface. The reason being is that we need to give it some place to actually form and blow out. We want to make sure that the most active portion of our plasma jet, the bright purple portion that's just right outside the nozzle itself, is getting a good amount of water vapor entrained in that plume, such that we're also getting a lot of hydroxyl radicals that are forming. With all the end stage production in the water and such, you're talking about peroxynitrous acid formation, hydrogen peroxide. That's where a lot of the ORP value ends up coming from, that oxidation reduction potential.
Yeah.
Are you worried about, Ryan? As the water heats up and you get evaporation, 15 minutes sounds like a long time. So that 4 millimeters is going to change over time, yeah?
Yep.
Not so much. With this configuration, most of it was a lot of spraying and dripping and such, but our final volume, we lost about 50 milliliters to evaporation. So there was a little bit of a gap change by the end, but it's not significant. So.
Yeah, we used to also have the same issue. We used to just put it right on top of the water, but then, as Christian said, that gap began to increase. So what we did was to just dip it in and then just whatever we can. So that's what we do here.
Yeah. Sure.
But then we also, so the flip side to that is, I think that's one of the reasons why one of our jets, we already burned through one jet and then plasma treat on a Sunday overnighted a new jet to us.
Oh, really?
Extraordinary customer service. Okay.
Well, glad to hear that too.
Unbelievable service. But I think there's an, and you're using DI here. If you're using tap water, I'm wondering whether there's a increased risk of corrosiveness if we use tap water and dip it in. So maybe there's some down the road there. I don't know what vital fluids are doing, but maybe there's some way to, I don't know, have a tip that could be less corrosive or something, and still resist the heat. Or I'm not sure what, from a manufacturing standpoint, what tricks could be made there.
You're absolutely right. The reason for starting with distilled water is just to have a very consistent starting point. And you can see we didn't have quite the same sample volume that you had, but even over 22 samples, our standard deviations are very low. It's very reproducible from this particular standpoint. The tricky thing about with tap water is, wherever which tap you get it from is going to be different. So most of the US, for example, we have a lot of chlorinated chemistry in the tap water. That will end up causing quite a bit of problems as well for the materials of the electrode and such. That nozzle tip, plasma treat has several other material choices that are more robust to these kind of conditions as well. So it's something that we could discuss further with you for sure, and we can try and get those into your hands and let you guys have some fun. But in general, we've run this particular tool in water for hundreds of hours at this point, it seems. So without even having to replace the nozzle. So you're probably right, where tap water is probably the biggest culprit in this case. So. But the other thing that I wanted to kind of mention for the sake of, if I'm producing this in, Silicon Valley here, our grower is about 2 hours north in California. How do I get the samples to them at a reasonable time? So we did a little bit of a stability study over the course of about a month. Obviously there's that very, very fast drop off, meaning like within the first few minutes of certain unstable species like that hydrogen peroxide from hydroxyl radicals and such. But if we give it, 5 minutes, take our test to get a starting point, or T0, if you will, But then kind of moving out over days, weeks, whatever have you, there's general stability in the solution. So if I pack up a couple growlers of this solution and put it in a box and ship it overnight to our collaborator, it's going to be the same thing by the time she gets it, and it can sit on the shelf for when she's ready to use it over the course of a few more days, a week or so, as she continuously waters the plants. So as for the actual study, day one, get started with the clippings from the mother plants. Usually they're about 6 inches tall to get started. 45 to 50 plants each. They're in separate trays with this rock wool, which is a inert media. The rock wool itself is actually moistened or heavily wetted, soaked up with 2 different solutions. Our control solution was her filtered tap water, along with this common nutrient supplement for this particular time, which is Clone X, a cloning solution that normalizes out at about pH 5.6. Our activated water group, we were shipping to her a diluted sample down to about 3.2, 3.3 pH. She was adding the same Clone X and then a pH up solution. So the reason for this being, we found that if we shipped the 2.3, 2.5 pH, it is too acidic for the plants. The plants are happier somewhere closer to 5.6 to 6 pH.
Ryan,
essentially it causes.
Can you legally ship something that is 2.5 pH?
Good question.
We are worried about that.
Yeah.
I don't think FedEx and those other. So we have a little bit of intel on that, and we think there's a Neil, was that what did you pick up on?
From what I recall from Andy, the threshold was actually a pH of 2 and not 2.5.
Okay.
So also through some of our contacts that we have with FDA and such, PAW is considered grass, generally regarded as safe. Meaning there are no regulations usually around it. It's more a problem of shipping a liquid than it actually being a acid in this case. So it doesn't surprise me that maybe they have an acid level that has problems. We put some fragile, do not throw kind of stickers on the boxes as we shipped them, but it's not something that was it caused any issues with shipping and stuff. And we had talked to a couple representatives from UPS, for example, as well.
Okay, good.
But that's a good note as well.
What kind of containers are you using for keeping the water or shipping the plastic glass?
No, it's somewhat similar to what you would get if you had ordered from Sigma Aldrich or something like that. We had glass bottles. They were sealed up with parafilm, sealed the caps onto them and such, bagged them, as you said, but also put an absorbent and padding material around them as well to make sure that they were handled properly. So, but it was shipped, spring, summer. So temperatures anywhere from 60 degrees up to the 90s, during shipping as well. So a little bit of variability in that kind of overnight transport as well. Okay. So essentially, swapping out filtered water for PAW, very small amount of buffer to add it to get to the same pH between the 2 samples as well.
Ryan, I'm just gonna say, I think I gave you 10 minutes, but we're making a lot of comments, so feel free to just take as much time as you want. Okay, you're not.
Okay, all right, thanks.
You're not under a 10-minute restriction anymore. That would be very unfair.
No, I appreciate all the questions too.
Exactly. So this is the kind of conversation we want. So screw the program. Yeah, keep going.
Okay. So up to this point, up to 5 days, they were just soaking up the moisture that was in the rock wool. At the day 5, which is the typical for her control set, started foliar misting of these solutions. So again, it's the same solution that was put into the rock wool is now being misted from above. So that's a kind of comment towards, can the PAW be used in sort of an irrigation system? 10 days in, we start to see algae growth on the rock wool, consistent both between the control and the PAW, which is good because, again, the original solution that we had shipped them was about 2.5. There was 0 algal growth, and actually it didn't, do the plants any good as well. So the pH was a big factor there. Day 12, she noted that there were 3, 4, 5 plants from just the PAW tray that started to show white nodules, which is the root growth forming actually above the rock wool itself. None were seen in the control group. We didn't actually start to see those root growth until much later in the control group, but some additional counts were noted by day 18. By day 21, as she noticed, that the growth of the plants was getting about to the height above the rock wool was about the right stage to start, looking at transplanting them. And this is where, she had checked underneath to see the root progress between the control group and the PAW. And she noted that there was, a significant dent. It's a little bit hard because of the porous material on the underside, but there's a significantly larger and denser grouping of root growth at the PAW group at the same time point. And then just the following day, when she was ready to start the transport or transplant to soil, took this final picture that kind of goes with, what Nansen had showed, previously as well. The control group, sure, looks nice and healthy, but the PAW group looks much, much better. There's some significantly different growth there from that grouping. So,
is it possible that the PAW was non-uniformly added to these trays?
It's possible. There's not she showed us some video at some point. I didn't have a solid picture that she had shared with us. The irrigation system is, some flexible tubing, right, basically. And it's it doesn't stay quite in place perfectly. Right. So as best she could, this was about as even as she could, but it is possible, that there was some non-uniformity there. So, yeah. Yeah. So just some quick conclusions, PAW is stable for several weeks. So shipping it for this type of application is feasible. After, a handful of days, there's actually, already significant difference of 9 days between the control group and PAW group related to that root presence coming up there after in the rock wool. But at that 21-day point, the PAW group is, significantly enhanced, or further along than the control group at this point. Just some, additional thoughts going forward. This is only the first stage of these plants. It would be great to actually follow them further into like the flowering stage, and then maybe do some additional analysis to see if there's any sort of compositional change, to these plants later on. There's already a significant amount of post-flowering stage testing that these plants usually go through. So there's definitely a plethora of knowledge there as well. And then at least on the plasma treat side, we're also concerned with eventually upscaling the production of PAW for much larger operations, which we're already keenly capable of doing, because we set up arrays of up to 64 jets, so it's not unheard of for us to upscale in array structure from here. So that's what I have. I think I still managed to stay maybe about close to 10 minutes of speaking. But any other questions, I'm more than happy to answer. If you think of something a week from now as you're staring at your house plants, I'd also be more than happy to discuss. So feel free to jot down the contact information and reach out.
Ryan, this was excellent, and I just want to steal my own thunder here by saying that later on I will be presenting on some of the data that Patrice, my postdoc, has generated, and one of the cool things that this was entirely his idea was to count the trichome density on tomato plants with and without a PAW treatment, and he saw a significant increase in trichome density. And since we're talking cannabis here, this is something you, of course, maybe not you, but the producers of cannabis would be very, very interested in is the trichome density. So think of that as a response variable to quantify. Okay.
Yeah, that's a good point. Thank you.
Ryan,
Great.
did you counting trichomes in your experiment or no?
No. As far as I know, our collaborator kept the plants for personal use. The rest of them, her business is going through this cloning cycle. So I think she turns around and sells these clones for $1,000 a piece. So, and a lot of it's related to operational costs. There's a lot of electricity and, whatever else, humidity control. So she ended up keeping, since they're, experimental, parts, it was a shame to destroy them necessarily. So I'm pretty sure she continued on with them, but kept them for private use, we'll say at this point. So.
Right.
Hey, Ryan, Nate Ahlens at University of Minnesota here. Great presentation. I was curious about
Nate. Can you increase the volume of your sound? It's a little low in my end, at least compared to others.
I can't. If I talk really loud, does that help?
A little bit. Yep.
Okay. So looking at the picture of those clones you had, a really common problem that a lot of growers face is some chlorosis at that day 22, and it looked like your paw group actually had greener leaves in comparison to the control group.
Yeah, I mean, it would part of that, compositional analysis later down the road would be great to do that color analysis. I'm forgetting off the top of my head the analysis technique, but I've seen people do it with basil plants and a lot of other leafy green kind of things. So, sometimes I chalk it up to just differences in the cell phone camera that's used, maybe. And her lighting situation. So, but yeah, it's a good possibility. It's also kind of a denser canopy that's on there that we're seeing. But yeah, it'd be a good idea to go through that as well.
Right. Thank you.
And there are definitely ways to optimize that. So this falls into optical sensing, which we in my lab do a lot of. So this is one of the reasons why we are carefully imaging these plants, precisely for that reason, to quantify the potential change in or difference in greenness. Excellent. Well, Ryan, thank you so much. That was great. If you will unshare for Paul, we will hand it over to you.
Yeah. Okay. Thank you. I will start sharing my screen. Yes, I think you can see it now.
Yes.
We can.
All right, that's good. Yeah. All right.
And if you go to slideshow mode, then it's even better.
Yeah, you can see the first slide now, right? Can't you? Yeah.
Not in slideshow mode.
We see kind of a split screen where we see all of your slides on the left side and then one of them.
You should at the bottom right have a little icon that can put you into slideshow mode.
I cannot find that, unfortunately.
Okay.
I'm sorry about that, but I think I'll have to do it this way then.
That's fine. That's fine. We'll work with it.
Okay. All right. Well, I'm from Vital Fluid. Vital Fluid is a high-tech company specialized in plasma technology. We are a spin-off of the Technical University in Eindhoven. And what we do is we develop equipment to produce plasma activated water. And we also develop, in close cooperation with end users, applications of plasma activated water. So our company focuses only on plasma activated water, and we develop applications related to agriculture to grow crops more sustainably. Here you see a fertilizer factory. I think there are approximately 600 to 700 of these factories worldwide.
Paul, your
Sorry,
slides are not moving.
Yeah,
Sorry?
Your slides,
We just
they're
see your
not
first
moving. So
slide.
we're still just seeing your first slide there.
Oh,
yeah.
So
What happens that when you go for a slideshow, it creates a second file. So
Right.
if you are sharing the first one, you're not seeing the presentation itself.
maybe
You need to share the second thing.
right.
then I will just stop it. Can you see it now?
Yep.
Yes, we can.
All right, you
Yeah.
can see it.
Try to switch. Try to just go to the next slide, just back and forth, just so we can see that it actually changes.
Yeah, now it changed.
Now it changes.
Okay.
Yeah. Okay. Okay.
Sorry
Now
about
you're
that,
good.
but okay. As I mentioned, this is one of the I think 700 fertilizer companies in the world. They produce nitrogen fertilizer, and they use actually fossil fuels by that. So the process is driven by fossil fuels. We want to offer an alternative for this process, and then not use fossil fuels as for driving this process, but we use electricity, we use plasma to fixate nitrogen. And this is another familiar picture, I think. Here you see pesticide being sprayed on crops. And this is the second application we at Vital Fluid are working on. We want to offer farmers and growers a sustainable alternative for chemical pesticides. But okay, we need to be fair. We need those chemicals. We need nitrogen fertilizers, and we need pesticides to feed the world. If these substances weren't there, then half of the world population wouldn't have anything to eat. But they come with a significant environmental burden, unfortunately. We work on plasma activated water, and as you all know, the inputs are only water, air, and electricity. And if you treat water with plasma, then the water obtains temporary disinfecting properties. And these disinfecting properties can be used, for instance, to spray it on crops, to keep them clean, to keep them healthy, and to prevent the use of chemical pesticides. And that is the most important application we are currently working on. And yeah, the beautiful thing with plasma activated water is that when you leave the water to rest, after a while, the disinfecting properties will go away. What remains is water with an elevated concentration of nitrate, and nitrate is the most important fertilizer for plants and crops to grow. The system you see here is our lab unit. We have done a lot of proof of concept testing with these units. This unit produces half a liter of plasma activated water, and we have been testing in medical applications, wound treatment. We have been testing in water purification, but our main focus lies on agricultural applications. And if I go to the next slide, I hope this will work. Yeah.
Yep.
Okay. Yeah. Okay. That is plant nutrition. Here you see our Freya system, and this Freya system, what it does basically is takes the nitrogen out of the air, split it with plasma, and then dissolve it into the water in the form of nitrate. And the water that comes out of this machine doesn't have any disinfecting properties. It is water only with an elevated concentration of nitrate. Concentration is about 300 millimoles per liter. And that, of course, can be used in feed water. It needs to be diluted. With one machine, you can supply approximately 1 up to 4 hectares of cultivation with nitrogen fertilizer. So, of course, what comes out of the machine is only the nitrogen fertilizer. Crops, they need a tailor-made fertilizer recipe containing all the nutrients, and nitrogen is just one part of that. So our machine produces the nitrogen fertilizer, which basically is about 60% of all nutrients that plants need. But you need to mix it with the other nutrients before you can use it as feed water in drip irrigation system. There was a question about if it's possible to use plasma activated water in drip irrigation. Well, that's what we do. That's what we do with our technology and machines. We have currently running 10 units, 10 Freya units in the US. You can buy actually lettuce in the supermarket in the US that has been grown with our machine with plasma activated water. But that is only, of course, the fertilizing properties of plasma activated water that is used there. We are also having running projects in Spain, 2 projects in Spain, also for the fertilizing application. And there is a tomato grower in Morocco which is using our systems to prove that the CO2 impact of their fertilizer can be reduced by using plasma technology, because when you use conventional fertilizer, the CO2 footprint is quite large, and with plasma technology, especially when you use solar energy or wind energy, the CO2 footprint is much lower.
Paul, a quick question here. Are any of those companies able to tap into governmental subsidies? Kind of like you buy. So I drive a hydrogen vehicle. I'm one of the few that actually has a hydrogen vehicle in California. But even if you buy an electric vehicle, you get subsidies. Are there any, by going this route, are there any opportunities that you are aware of that these companies can, and other companies for that matter, could benefit from subsidies or something by going green this way?
Yes, I think so. With the companies in Spain and Europe and Morocco, we are testing. Those are subsidized projects, but I think I know that there are possibilities to, when they want to buy these our equipment for these purposes, then there are possibilities to get subsidies on the equipment. Yeah. All right, so that is one application we are working on. Another application, and as I mentioned earlier, this is our main focus, is plant health. Because if you use the temporary properties, the temporary disinfecting properties of plasma activated water, you can actually keep crops clean, healthy, and fit. So it's more difficult for viruses and microorganisms and fungi to infect them. I think it was 2018, 2019, we started some small scale tests with a rose grower here in the Netherlands, because he wanted to get rid of the chemicals he's using at this moment. And we started our first trials in powdery mildew in roses, and we saw quite some interesting results. So we decided to set up some more scientific tests on that. And the first test was to see, what effect does the dose have on powdery mildew? So we produced different types of plasma activated water and treated leaves that were infected with powdery mildew. And we upscaled this in step 2. And again, we saw some quite good results. And then after that, after we found the right dose of plasma activated water that could be used, we went through a small scale testing in the greenhouse itself. It was an area of approximately 10 square meters. And we have in 2024, I see that video is not running, so I will start it now. Here you can see an actually spraying robot. The spraying robot is spraying plasma activated water on the roses in the greenhouse itself. And we have already proved that plasma activated water performs better, has a higher efficacy than the chemical control. And so the synthetic pesticide doesn't doesn't work any better than our plasma activated water. And the beautiful thing is plasma activated water, the residues it leaves on crops, it's a fertilizer, so it will be eaten. There are no residues. Well, if you use the synthetic chemicals, there of course a lot of residues left on the plant.
Do you have any idea of the mechanism here? Do you know what the fungicidal properties are? Have you been able to break it down or try to experimentally test whether it's the pH, the ORP, the conductivity, any ideas there?
Yeah, well, the reactive species in plasma activated water, they oxidize, they have oxidizing properties. So if you have a microorganism, then the cell membrane will be attacked by these oxidizing properties species, and then you will have lesion of the bacteria or the virus or whatever.
So you think it's the hydrogen peroxide that is doing the job?
Hydrogen peroxide, peroxynitrate, nitrate, those kind of compounds. Yeah, those are responsible for the disinfecting effect of plasma activated water.
Okay, Paul, I have a question. So, is there any impact on the colors of the plants? For instance, in the roses you're showing, because depending on the pH, the anthocyanins that are responsible for color can change. Color, do you see a difference?
Yes, of course. We do, before we go into these large scale testing, we do phytotoxicity test. We do a specific test with different formulas and different recipes of plasma activated water. You also can add some surfactants to make sure that all the leaves are covered with plasma activated water. So there is a lot of research going up front before we do these kinds of tests. And if we see any damage, then of course the grower says, well, oh, that's nice, but I'm not going to use that. So that's already been tested before we go into the field. All right, we also did some testing on powdery mildew in cucumber, and well, we saw again some very interesting effects. And first, of course, we analyzed the crop activity, and we determined if plasma activated water was indeed active against mildew in cucumber plants. And here you can see some very nice results. I hope you can see it, because there are 4 pictures on the right of the graph, and the top one on the left is untreated. The top one on the right is the reference, so that is what growers are using at this moment, and you still can see some white spots on the leaves. The bottom 2 are plasma activated water. So you can see here also in the results that plasma activated water works better than what growers are using at this moment. So also for cucumber, we are setting up full scale trials at this moment to further develop this into an application that growers can use.
Maybe I missed it. What
We've
was
also-
the application frequency?
The application frequency was about, I think, for cucumber spraying 1 or 2 times a week, and then it is 1,500 liters per hectare.
Okay, thank you.
Yeah, and when you compare that to the synthetic pesticides which are in use, then well, they need to spray as much as plasma activated water. So same frequency. Yeah, well, the price, of course, the business case, those chemical pesticides are quite expensive, and producing plasma activated water, well, it costs energy. But we think we have a very interesting business case there for growers. Okay, well, we also did some testing in orchids. Ventura is also a fungi. Again, we tested it on crop safety and efficacy, and well, then we started to do some small scale, well, let's say some quick and dirty testing. And we didn't use any optimized plasma activated water. But again, we saw some very interesting results, and we are at this moment running trials here in orchids on a very large scale. So, we have also been talking about seed treatment this evening. It's evening here in the Netherlands. We have been working on that. This was the first application we started to work on. We cooperate with the bigger seed companies in the world. Seeds, of course, need to be clean, need to be disinfected. You can use pesticides for that. You can use chlorine for that, but you can also use plasma activated water for that. So that's what we are doing. Well, we see some very nice results there. If you see here, for instance, the chlorine treatment, well, PHW gives some better results than chlorine. So we have something interesting here. We have been testing application of this on vegetable seeds, and we are now cooperating with Pest, a very large seed treating machine. They are building seed treatment machines, and we are looking if we can scale up this application to field crops, to corn, to wheat, to large amounts of seeds. So that's what we are working on at this moment. The last application I want to show you, we are working on, based on the disinfecting properties of plasma activated water, is the post-harvest treatment. We have been treating apples and pears with plasma activated water, and we have given them quite some extreme exposure to quite strong plasma activated water, which of course you can make it really, really strong, because yeah, if you keep activating the water, what you will get is nitric acid in a very high concentration. But again, we have seen that the apples, well, they have very little damage to no damage after exposure to plasma activated water. So what we are in follow-up testing, we are doing the disinfection test, shelf life test, to see if shelf life can be enhanced by using plasma activated water. Concerning the development of our technology and our machines, here again is the lab unit we use for concept testing. We are still using that, of course, because there are a lot of parties and universities that are interested in to work with plasma activated water to see what possibilities it has. We have upscaled this unit so it can produce 2 liters of plasma with disinfecting properties, plasma activated water per hour. Our fryer system produces approximately 500 liters per hour, so that's quite a lot, and the concentration that comes out is 300 millimoles per liter nitrate. So you have to dilute it when you use it, when you want to use it as feed water. And what we are working on at this moment is what we call feeder, and that system is specifically being developed for disinfecting properties, because we want to extend shelf life. We want turn on knots to optimize the composition of plasma activated water. More peroxide, less peroxide, more nitrate, less nitrate. We want to control that, and that is what we can do with our feeder machine. This machine will become available at the beginning of 2025. We have it in the lab already running and doing some tests with it, but it will become commercially available in 2025. All right. Well, that was my presentation for today.
Excellent, Paul. Thank you very much. Any additional questions for Vital Fluids at this point?
Yes, Paul, thank you for your nice presentation. I'm concerned and thinking about in terms of applying directly on food products. How is it in terms of regulation? How have you been dealing with that, or have you tried some kind of toxic analysis or something in that direction that you want to share?
Yeah, well, we have done some residues measurements also on crops, also on cucumbers, the produce itself, the cucumber leaves, and we see no effect of the plasma treatment. So there's not a higher nitrate or nitrite concentration or whatever. But you are right. If you want to use it as as water to treat produce to enhance shelf life, then and it has disinfectant properties, then it's called a biocide. Here in Europe, we have quite some severe legislation, and you have a biocide regulation and you have a regulation for crop protection. Takes quite a while to get through that process, but we have started these procedures, so we are working on that. Fortunately, the rest of the world is less severe. There, a procedure for admission of your product for crop protection and also as a biocide takes approximately 2 years, and we also started those processes.
Okay,
right.
Thanks.
You need to play golf with the right people, Paul.
Yeah. Yeah. All right.
And if I could just step in for just 1 second to make an additional comment. Christian, you had asked earlier about which components of the PAW, does really well for the powdery mildew and such. At least from the biomedical application side of whether you are directly plasma treating something or producing plasma activated water and then applying it. We've seen some, the way they've tested it out is, you could do direct plasma treatment of bacteria, viruses, spores, fungal, sources, or, produce PAW in this case, or just nitrogen fixation produced from the plasma activated water. So producing plasma using the gas and bubbling it through. In this case, what they found is that just the nitrogen fixation portion, so getting that pH low, works just fine against bacteria, for example. However, when you get to something that's a little bit more resistant to aggravation in a system, meaning something that's fungal, something that has spores, even sporulating bacteria, you either need direct plasma treatment where you have direct access to like bombardment of the microbes, or you need to provide that oxygen, the oxygen species, basically. So hydrogen peroxide, hydroxyl, or ozone in this case. So a lot of times the best way to take care of these pathogens is to directly treat water, make sure the jet's impinging on the water itself to get those hydroxyl radicals, and then using that solution to even at a lower temperature like a dielectric barrier or a corona, just won't work out so well for that direct treatment of plasma. So I just wanted to add that comment because it comes from a decade's worth of biomedical research from from that other side.
Right. No, that's great. Excellent. Very good. Well, thank you. I'm not planning on breaks, so I'm assuming that people can just go out and take their own personal breaks. I think we should just plow through here, and it will be recorded so people can go back. But my thought was now Neil and Nick, you can take it from here. Is that okay?
Yep.
And talk about the survey?
Yep, sounds
Yes, good. Nick, do you want to share your screen?
good. Can everybody see my screen okay?
Absolutely.
Good.
Okay. Okay. Hey everybody, how you doing? So I want to give an update, or we're going to give an update on our survey, greenhouse and nursery plant awareness for plasma activated water for young plant producers. I'm not sure how many of you were on our previous call, but I'll give a review as we go through it. The purpose of this survey is intended for growers, farm operation members, owners of operations that produce and use young plants. This was to gain insight into grower practices, potential issues, specifically looking at growers who have who use seedling and young plants in their production. And then to help us identify species of commercial importance that growers are potentially having issues with, and then use that information to target species in future trials. And then ultimately use this to gain insight into grower awareness and how much is plasma activated water being used in production, and who can we target with this research moving forward. And really just looking at, I'm going to look at these two terms. I want you to kind of pay attention to them, is willingness to learn and willingness to try. I'm just going to have this little star here. These terms are going to come up periodically throughout here. So you're going to see WTL and WTT. So that's what they mean as we see it. And so really understand is, how much does this willingness to learn and willingness to try vary among awareness of PAW and the characteristics of these growers in terms of their farm operations, what they grow, et cetera, et cetera. So let's talk about the survey. We sent out a survey request emails to about 100 sources, mostly, horticulture media, trade journals, and then as well as green industry associations, such as various state landscape and nursery associations, Plant California Alliance, greenhouse grower associations. We sent out English as well as Spanish versions of this survey, and a total of 153 individuals participated in the survey. That being said, 82 of them fully completed the survey. And then here's just a snapshot of individuals within the US and Canada who participated, just kind of give a good spread geographically where they're located. We had a handful of individuals in Europe and Asia who participated as well. So the content, the participants were asked, how willing are you to learn about the application of PAW? How willing are you to learn to try about PAW within your operation? And then what are the details about your farm operation characteristics? What is your growing environment? How big is your production area? Do you grow in a greenhouse? Do you grow in a nursery? Do you grow what crops do you grow? What are your production methods? Are you conventional? Are you organic? Are you a combination of both? If so, what combination? Do you use seeds or seedlings within your production methods? How often do you encounter a lower germination rate than that on the label? How often do you encounter poor seedling growth? And do you use pre-planting seed treatments and treatments to improve your seedling growth? So to go through these, 78% of participants showed an interest in learning about PAW. 55% showed that they are willing to try PAW. And then I'm not going to go through all of those because there's a lot of different questions we asked. But these survey results are summarized into our newsletter that's publicly available on the website. This is Christian's website. It is specifically volume 2, issue 2. I had a QR code. I'm not sure why it's not coming up. As well as the previous cold plasma meeting. There's all those survey results. And then here's that QR code that's going to go directly to that newsletter to show the summary of those results. So I'm going to do a deeper dive, talk about our results here and what we did. So we really want to understand was what factors do your farm operations influence these users' willingness to try, willingness to learn about PAW. Again, there's that. And we use what's called a probate regression model. I'm just going to glance over this really quick. Where we did our dependent variable was if you're willing to learn about it, if you're not willing to learn about it, et cetera, et cetera. And as well as all these different independent farm operations characteristics if you use seed treatments, pre-planting seed treatments, et cetera, et cetera. And how does that change? So if we look at our farmer operation characteristics and willingness to learn, we saw a really nice positive correlation with indication that these growers would be more willing to learn with your production area. So if you had a larger area, they were more willing to learn about it. Greenhouse, so 78% of the individuals use greenhouse in the production. So if you use greenhouses, you're more willing to learn. 25% if you use herbs, you're more willing to learn about PAW. It was 25% of the individuals surveyed. And then as if you use seed in your production. Willingness to try, if we saw a really strong positive indication that they'd be willing to try, was if they used herbs within their production, not produce, and if they grew in an indoor vertical farm. These are the percentages are how many people responded who actually did that. So not the majority of respondents, but a portion.
Do we know, Nick, if any of them were cannabis growers?
There was no indication of cannabis growers, but that was also not a
target group,
group or an option that we included. So there was others, and there was a question about cannabis, but nobody indicated they were solely cannabis growers. I did email the survey to one cannabis association group. I don't know if it was disseminated. Right. So what had negative?
Ryan, do you have? Is there like a network of people? I mean, we're gonna do this survey again. Are you connected with anybody that down the road? Because we are gonna, and Nick will talk about this later on. But can we send this out? Is this something that, and even this video here, can you send it out to some cannabis growers?
Yes, I think there's a good opportunity there. I can try and connect everybody together, maybe offline, at some point.
What about you, Paul? In the Netherlands, you must be growing some weed over there as well.
Yes, they are actually growing, and it's getting more and more legally, but we are not involved in cultivation of cannabis at this moment. So.
And there could be trade journals like Cannabis Business Times is a popular trade journal that has a weekly e-newsletter. So I bet they would help disseminate information.
Yeah, I think we did send it out to MMJ Daily, which goes out to a large portion. I'm not sure how many responses we got from that. So there's opportunities to cast a wider net to capture more of the cannabis community moving forward. If the more that a user reported lower germination rates than what was on the package, the higher that they would be willing to try PAW. It makes sense, right? We want to maximize as much germination as possible if they're paying for these seeds, especially expensive seeds. So in summary, this information can really be used to target marketing strategies that'll help facilitate adoption of PAW among different sectors of the agricultural industry. It also really helps us as a research group target specific individuals to work with and crops to work with. So farmers with larger production areas are more willing to learn about PAW. The same goes for farmers who use greenhouses or indoor vertical farms. But the opposite was found for farmers who use outdoor container nurseries. So this is, it's indicative of those with having greater financial capacity to adopt new technologies such as PAW compared to smaller farms that have smaller margins and might not really want to adopt these until they see more proof. So future directions, where are we going to move from with this survey? We are currently preparing a manuscript, a peer-reviewed manuscript for publication. So you should see that pretty soon. That'll have a wide variety of this data. And then we also, at the conclusion of this project, we're going to conduct a second version of this survey. And really what the hopes is, as we start to disseminate more research, more results, how does this awareness or adoption of PAW change within the community? Hopefully we see more. And so we're going to do it again. So be on the lookout for that. yeah, any questions?
Questions, and I don't know, I dropped out, the connection dropped, but were you all able to see it?
Yeah.
Yes,
Yeah, it's switched to Anil.
Fantastic. Great.
maybe.
All right. If there are no questions, let's hand it over to George in Minnesota here and give us an update there, George.
Okay, can you guys see my slide?
We see your background.
We can see, there we
Now-
go.
Now, yes.
Good. I was worried there for a sec. Okay. All right. So I'm George here at the University of Minnesota, and I'm presenting today on some updates. This is not everything we do. My team, oh, okay. So this is the team in Minnesota. So of course, myself, Nathan joined us. And then we have Ase Masume Rashid is a postdoc who joined us recently and has been feverishly working on putting 2 manuscripts together so far. I have Grayson, who is an undergraduate student working a lot also on cold plasma in my lab. And recently, Cecia Flores joined us. So what do we do? This is not the only thing we do. My lab is a lab that concentrates on grain science and grain quality, but I started working with cold plasma because I was interested in how it will impact foods. But before we kind of dive into that, I was really interested in understanding how PAW changes when it is treated. And for sure, there have been some data already presented here. So I'll skip on some of the things I'll present, but also we wanted to we were also interested in investigating how PAW can be used to germinate cereal grains. This is very important for the brewery industry, but for today, I'll talk about just the water part. So this is the system I use. Is from a company called Enercon in Wisconsin. So Ryan, I wasn't able to buy the unit from Plasma Tree, but I obviously did recommend you guys to Christian. So it's basically a blown ion plasma that produces positively charged electrons or ions. And this is what we use to treat most of our samples in my lab. This here is a picture of the system, and that's Ase, my technician. And as we were discussing earlier, when we're treating water, we actually dip it in the water. We haven't had any problem so far. So we'll continue to do that because we were having issues with the height of the plasma jet on top of the water because there was evaporation. So for this presentation, I'm going to talk about some work we did. This is just a small part of it, but basically we wanted to understand how water changes when it's treated with cold plasma. So this is important to me because when you look at literature, there's a lot of information on the pH, conductivity, and all that, but we wanted to dig a little bit deeper into what really goes on. So we looked into the minerals. We actually analyzed over 25 minerals, and I'll present just a few of them. So what we did was to take city water and tap water, and we did this because we shouldn't only look at pH, conductivity, and ORP, but also look at the minerals. It is obvious that DI water and tap or city water will be very different when it comes to the minerals, the amount of minerals in these 2 different types of water. The other thing we were interested in is, are the water sources from Minnesota, UC Davis, and Cornell different? And not only are they different, but when you use it for PAW, would you get the same water in terms of its chemical characteristics? So what we did was we took 1 liter, and then we treated the water at 5, 15, 30, and 60 minutes for tap water and distilled water. The distilled water was mainly for Minnesota, and the tap water was from the 3 different locations. So this is basically what we did. Now, the data you see here is showing you how much water we lost from the 1 liter that we started with. So it's not significant, but it's still a lot. And I actually liked the system that Plasma Treat had, where they had this plastic container that prevented loss of water. I think I would be looking into that. So I'll dive straight into the results. So we'll start with city water versus DI water from Minnesota. So it is obvious from the two different types of water that the DI water is the black one, that irrespective of whether you use DI water or city water, you get a decrease in pH. Initially, we saw some differences, but ultimately, as treatment time went on, they kind of became the same. We got pretty much very similar pHs. When we look at ORP, we started with very different ORPs, but again, as treatment went on, we expected an increase, and they were also very similar. Conductivity, however, was different for tap water. Tap water was higher than that of deionized water. And this also plays a lot in the amount of minerals. We always used to think that it is the nitrate and the nitrite, but I think the minerals also play a role in the amount, in the extent of conductivity, increase in conductivity that you get. Total solids was also similar to conductivity. So we obviously saw that the tap water had higher total salts than the deionized water. Okay. And then when we look at the nitrate and the nitrite, we see that there was a general increase in nitrates for both DI water and tap water, but it was significantly higher in the tap water than the DI water. So they are not the same. And I see publications where most people start with DI water because they want to have some control, which is good, but it is obviously clear that using tap water results in higher nitrates. When you look at nitrite, we see that the initial change in nitrite in the first 20 minutes or 30 minutes of the treatment was significantly different. We had tap water had significantly higher nitrite, but then they all began to drop. At the end of 50, 60 minutes, we had pretty much the same amount of nitrite left. And we know that the loss in nitrite actually is contributing to the increase in the nitrates. Ammonium nitrogen is another parameter we measured, and it is obvious from the tap water that it had barely any ammonium nitrogen. City water from Minnesota had about 2.2 milligram per liter of ammonium nitrogen, which significantly reduced. Now, for me, this is important because too much ammonium nitrogen is actually toxic to to plants and also to microbes in the soil. Now, ammonium nitrogen is known to be higher in underground water and is obviously coming from a lot of the runoffs from the fertilizer that I used. So when I saw this data, I saw another opportunity. We could potentially use plasma to treat wastewater to reduce the amount of ammonium nitrogen in some of these water samples. Now, irrespective of the fact that we got a significant drop as treatment.
George, if we did that, where would that nitrogen go? Would that go into the atmosphere?
So that is the question. That is what we don't know now. I believe some of it will go into the nitrates, but there are chances that some of it will just be nitrogen gas. Yeah,
which wouldn't be good, right? So we would need to capture that in some way.
Yes, for sure.
And these ammonium levels are very good. Usually I'm looking for, say, less than 30 parts per million in a nutrient solution.
Yeah, so that's good. But just the fact that it was able to significantly reduce it. So if we have water that has more than the 30, then we could potentially use a plasma to significantly reduce that. And the big difference, which was expected, of course, came from the hardness. DI water had barely any hardness because, of course, most of the minerals that causes the hardness were removed. But when we look at tap water, we see an increase in hardness. We saw that also in DI water, but it was more obvious in the tap water. So if you are using PAW for an application that requires some calcium or some minerals, you probably will be better off starting with tap water because most of these already come with some of these minerals in there. Then let's look at the mineral for calcium, for magnesium, for sodium. And these are just a few. As I said, we looked at about 27. We saw that the deionized water barely had any, and that was expected. Calcium seems to be significantly going up for the tap water. And again, this is very important because for food application, sometimes we need calcium for certain enzymatic reactions to actually proceed better. And this is very useful when you are making beer. So I'm not saying we should use PAW to make beer at this point, but we obviously can be able to add calcium to water and use it for certain applications. Potassium was the same, general increase in potassium. We see a much bigger increase in potassium in the deionized water compared to, in potassium compared to calcium and magnesium and sodium. And I'm still trying to grapple where that potassium is actually coming
George,
from.
have you,
Yes.
can you just, this is treatment time. When were these readings? You may have said that, but I think it would be good to reiterate. When was these readings performed? Was this 24 hours or 48 hours after treatment, or what? What time point are we looking at here?
Yeah. So for the pH conductivity ORP, all the analysis were done after 24 hours. Because earlier studies showed that from our lab shows that there's a significant change in those parameters. Essentially, that water is not very stable within the first 24 hours. For the minerals, after the 24 hours, we now took the samples to the water analysis lab, and I think it stayed with them for a couple of days for them to do the analysis. But we still saw all these differences.
So this is the problem we're having, right? We want detailed analysis, but we can't do them real time. It takes time. They need to be sent to an analytical lab. So we don't know exactly when each of these samples were analyzed. Is that correct?
Yes, but it still gives us some understanding on how
absolutely.
Yeah.
Right.
So now let's go to Minnesota versus
Excuse me, could I ask
California
a question?
versus...
I'm sorry,
right,
Ken Bradford at UC Davis. I'm curious, this is water being aerated.
Mm-hmm.
How are you getting absolute increases on all these other minerals?
So that is the big question that we are trying to answer. So we think that, so if you look at this, the distilled water versus the city water, we think that some of these minerals may already be in the water, but bound in a certain way to other chemicals that are not making them being identified as calcium. Or it could be that the PAW is, I don't want to say taking it from the air, which obviously we don't have calcium and potassium in the air, but I think whatever chemical reaction is going on in the water is causing these minerals, which inherently, in my view, were already in the water, being freed up to be picked up. That's what I think.
It's a closed system. It's a closed vessel.
Yes.
Based on what I saw today, that the water is a closed vessel. Another question. I noticed, I just looked at the earlier recording the other day as well. There is some water loss through this system, evaporation apparently. Have you corrected for concentration due to evaporation in these calculations?
So that's a very good point. We haven't corrected for that because the amount of water that we lost in our view was very small. The percentage was about 10 to 13%, which could actually be, you actually bring a very good point. So we will correct that, but we still see a slight increase, which I'm not sure where this is coming from. But yeah, we will correct that to get this a normalized. That's a very good point.
I noticed earlier there was data that salinity increased and so on. I really don't see where you can get new minerals from somewhere, from nowhere.
Totally agree with you.
I think that's a very, okay, thank you.
So Minnesota versus calcium versus California versus New York. Okay, so the idea here was to look at the question of, can you just take water from anywhere and produce PAW? Obviously, yes and no. Now, here we have water from Minnesota. This is city water from Minnesota, UC Davis, and Cornell. And we treated these samples the same way. 1 liter, we treated with plasma activated water. And obviously, as we expected, pH decreased for all of them. But the decrease in the pH was different for the water from the different sources, especially within the first 30 minutes. Now, as we continued the treatment, we saw that, especially after 60 minutes, the pH of all 3 water samples was the same. So it is important for you to know how long you want to treat your sample, because then if you're using water from different locations, it is going to be different. The PAW that you are going to get is going to be different. The same thing was seen for ORP, general increase in ORP, but significant differences in the first few minutes of treatment. Here we can see within the first 20 minutes, the water from Cornell actually had the least ORP to start with. And healthy water, when I check literature, healthy water should have an ORP between 300 to 500 millivolts. And it is obvious that the one from Cornell was way less than that. The one from Minnesota and California were okay, but irrespective of whether it was low or high, the treatment actually resulted in a general increase. Conductivity was the same, significant differences in the conductivity that we saw, especially after 60 minutes of treatment. Very, very different for the 3 different sources, with the water sample from Minnesota having the highest conductivity after treatment. We saw the same with total salts, and when we treated the samples, the one from Minnesota had the highest of the amount of total salt in these samples. Nitrate and nitrites were also very different, especially at the latter stages of treatment. The sample from UC Davis had the lowest nitrate levels when we did the treatment, at least for an hour, but we can see a very big divergence in these parameters, especially nitrate, after the 30 minutes of treatment. So the sample from Cornell had the highest amount of nitrates, followed by that from Minnesota and that from UC Davis. We saw a similar trend in nitrite, big increase in nitrite, and pretty much most of it was lost. But that change in the increase and loss was also very different. The sample from Cornell had the biggest of that change amongst the sample. Ammonium nitrogen, the samples from Cornell and UC Davis has the least ammonium nitrogen. So there's something actually going on with the water in Minnesota. Even though it is not as high as Neil said, it significantly had the highest ammonium nitrogen for the control samples. Hardness was very different as well. UC Davis water was the softest or the least hard. And when I looked at this data, I actually checked. So the official website for the city of Davis, they actually have the hardness of their water samples reported. And they were reporting that in Central Davis, water samples, they have a hardness of about 59. That is still pretty higher than what we got. So I'm not sure where the water samples came from, but it was really, really low in terms of hardness for compared to the one from Minnesota and also from Cornell. Mineral content, again, we see that increase in mineral content for Minnesota and Cornell, not so much for UC Davis. And you could see it's the same trend for magnesium, is the same trend for sodium. Potassium, however, I just cannot get my head around what happened here. But we saw that there was a general increase, but that increase in the one from UC Davis was much, much, much higher. So what do we learn from this? Well, the source of water for PAW production matters. We shouldn't just assume we can make PAW water from any source of water. Maybe it might work, but as we hone in on the applications for PAW, it is important that we kind of optimize the PAW for different applications. And in order to do that, we have to really consider the source of water that we are using. I would also recommend constant monitoring of the PAW that we produce because the water is changing so fast that if you assume that all the PAW you do is going to be the same, especially when you are using different sources of water, that might not be the case. And we do that in my lab, and I know Christian also does that in his lab. But the point I'm making here is not only the pH, conductivity, and the ORP, but also these other parameters that I have just showed. It may be necessary to precondition or modify the water prior to treatment or even after treatment, depending on what you want to use it for. This is not new, especially in the food industry. It is very common for brewers to actually precondition their water because the calcium content, the mineral content, or the hardness of that water could have an impact on alcohol production and also the taste and flavor of the beer. So these are the conclusions that I came up from this initial study. The second part of the study, which is the storage part, will be presented in our next meeting. We are almost done with the manuscript for this part of the experiment, and I definitely would take into account the comment made by Kent in the results that we will use for the publication. So with that, I will stop here for any questions or comments.
Excellent, George. And Kent, thank you for being here. This is it was a great comment.
Very great comment.
Yeah. Any additional comments to George before we move on?
One thing I can say about the UC Davis water is we took it from UC Davis in Briggs, not the city itself. So I'm not sure if the UC Davis did any treatment
Yeah.
to get into our lab building.
Yeah, it was really very different. So, well, if we are able to, once we kind of identify where you took it from, maybe that may help explain why that difference. Yeah, we're seeing those differences, but yeah, good point.
All right. I will jump in here and continue on from what I took away from your slides, George, which is that this is a very, very dynamic system. It is the water itself is, once it's treated, depending on how long we treat it, depending on the source of the water, we have a lot of going on in the water itself. And if we have some headspace, if we have some air here, and I should bring up immediately that Bruno, Miles, and George are key players in what I'm going to be showing here now, because this was part of a discussion we had in this great team we have about the importance of having some air above the and I don't know what vital fluids, what plasma treat, what you're doing when you ship water, when you we are finding out that amount of headspace there is kind of critical. So we're going to show some data on how that may impact the storageability and the stability of the PAW as we look at it after post treatment. So remember again what I showed earlier, and you kind of actually, even though George did not present that the same way as I did, there are some positive and negative correlations in the data that as conductivity goes up, then something else goes down. So there are these clear trends in the data, and we're definitely going to use that to model some of the characteristics of PAW and add minerals to it. And we are also, Kent, we share your astoundment. We are not exactly sure what the hell is going on here, but it's very interesting that we are seeing trends that are very unexpected, but we can model them and then we can start playing with them. So that's kind of where this goes from applied into more basic research. What I want to show here is some data that Miles has been collecting in my lab. So what you're looking at here is simply storing some PAW in some opened or closed containers. So you can see at the legend here, we have closed are sort of the solid line, and open are the dotted lines. And we have either DI water or we have the Davis tap water. And basically what you can see is that if you look at the conductivity, it stays somewhat stable, but maybe increasing a little bit over time. But then something happens after about 160 hours. This needs more replication, but it does seem like there's some stability over time. But basically, the deionized water has higher conductivity, which means that it has some of it has lower salts and other minerals in it. But we also looked at, and this is what we were really, really interested in, is what could we in some ways increase that stability by adding some bases. So we looked at adding either sodium hydroxide or, on some of the next slides, potassium hydroxide. We are particularly interested in the potassium hydroxide because if we could add some potassium together with the nitrate and ammonium, that would be making it a very, very interesting fertilizer. So what you can see, if I just re-show the slide before, you can see a big gap, a big difference between the two waters. And you can also see a big gap between open and closed systems. Well, that gap, that difference suddenly gets lower as we add 30 mil of 1 molar sodium hydroxide. So we get a much more consistent, much higher consistency, especially of our tap water, which is also very, very interesting. If we add sodium hydroxide, we may have somewhat lower conductivity, which is probably a good thing. And we can see that also now suddenly the two waters the DI and the tap water seem to perform very, very similarly. So very, very interesting in that regard. If we look at the nitrates, which is what we're looking at here, we are seeing that especially in an open system. So follow this dotted red line here. Within 48 hours, we are losing quite a bit of nitrate, right? So there's a large drop, and then it somewhat stabilizes. Well, we can actually, by adding, I'll show you here. So we get a load, and so we went from somewhere about 600 down to about 200 to 400. So we lost a lot of nitrate over time. But when we add sodium hydroxide to the mix here, we can actually stabilize that quite well and get even higher nitrate levels than we had before. So this is very, very interesting. We can get an even more stable system if we add potassium hydroxide, a little bit lower nitrate concentration, but very stable. So that's very, very interesting. We got that, and then we got similar levels for our DI and for our tap water. So this is really something we are hoping that Vital Fluids, Plasma Treat, and other companies out there consider as part of their operational system, is maybe after treatment adding different bases to stabilize their product.
And a lot of it has to do with the exchange between that peroxynitrous acid as it transfers from nitrite to nitrate, and also the interaction with hydrogen peroxide and those hydroxyl radicals that are temporarily there. and you're talking about over days in this situation. What is happening when you add a base in this case is you're actually shifting what is still an undissociated molecule, such that it actually can participate or not participate in this case with that chemistry, and it shifts the stability, that rate to a much slower rate at that point. So if you were to heat these solutions after the fact and essentially accelerate that process over the course of just an hour, and then you repeated the same study, you probably would it would have very little effect and very stable solution at the end of this. There's and this kind of goes also to kind of linking up with what George was showing earlier as well. If you're forming bases in the solution, the pKa value in this situation for these different plasma activated water solutions that you're producing, the stability of it and what's being presented or measurable in the solution is going to shift with the pH value, because with weak acids or weak bases, they're not 100% just the conjugate base or conjugate acid. So you kind of have to take that into consideration as well. Yeah. So George, I was gonna, kind of take that offline in conversation with you, but since Christian brought this up, I figured it was a good time to interject, and we're seeing more evidence of it right here.
Yeah, no, this is very, very this is a very good comment you actually brought in. And I was just thinking about the comment made by Kent, and I wanted to ask you a question as well. So don't you think, or don't we all think those calcium, potassium is probably coming from the electrode? If we use ceramic for any part of the electrode, we are probably losing those from the system itself, because it's obviously not coming from the air. So it's something to think about, and I've been looking into it. So I will just hand over to Christian, and just let's think about this. Maybe at the end we can brainstorm on this.
Both the internal electrode and the nozzle tip are both stainless steel in this case.
Stainless steel.
So you're talking about trace amounts of other elements. If I don't remember there being my metallurgy is much weaker than most other things, so I don't think potassium and such would be in there. But what I'm saying is, it might just be that it's there but not measured until a certain pH is measured. Yeah. So I forget from your slides, did you also measure those concentrations at time zero, meaning negative, basically? So prior to treatment, what were those values? Okay. All right. I need to go back and take a look at your slides then in the recording.
I can share the slides with you.
Okay.
Are you saying, Ryan, that we should consider heating up our PAW after treatment? Was that one of the comments you were making? Did I get that right?
If you want to force it to its end equilibrium state faster, yes.
Right.
But I don't think for a lot of our applications that we're talking about today, that's where we want it to go, because essentially we just have a nitric acid solution at that point, which is weak. We want to have it in this metastable region where it is still changing. It is still, moving around a little bit, but it's at a kind of plateau in its rate change, if you will. So some of the studies that I've done to kind of force the chemistry to, cooperate faster in the past is to heat the solution up. And it forces the hydrogen peroxide to work a lot faster, and or dissociate by itself a lot quicker and hit a stable point. If you wanted to test that theory, basically, is what I was saying. But when we go to actually apply it in any of these applications, that's the exact opposite of what you really want to do. You probably want to, and people are actually even freezing it and using it as ice cubes, or for like the meat counter for fish and things like that. it slowly, is available in liquid state at that point to keep, fish from smelling fishy, for example. Some of the studies to actually capture those first few minutes of the, post-production of the solution, people are freezing it immediately and then transferring it to an analytical lab and thawing just a little bit for that analysis right away, because you can actually prevent the chemistry from progressing up to that, after that point.
So Ryan,
Wow.
when we make the PAW, it gets hot.
Yeah.
So it gets hot. And what we realized was, and that's why Christian brought that point, was the first 24 hours, things keep increasing. It just keeps going up until it stabilizes after 24 hours. It could also be because after 24 hours, the temperature drops to a point where it is not changing that drastically. Of course, if you increase temperature, according to thermodynamics, chemical reactions change, increase and all that, but it was going to be very difficult to kind of control that first 24 hours unless you mechanically cool it down. And we just leave it. We just leave it for it to cool down before we start. So all the data that we are presenting here were collected at least 24 hours after things have settled down.
Yeah, the most volatile, if you will, portion of time is that first 24 hours. I'll agree with you on that. And then kind of keeping it at room temperature or sub-room temperature, it, kind of keeps it the most stable after that. Now, the reason for that increase that happens a lot of times is, again, because of that mixture of strong acids and weak acids. And over a certain amount of time, that nitrate transitioning to nitrate, you're transitioning from a weak acid to a strong acid. So the nitrite almost doesn't contribute to the pH value that you're measuring. It's almost 100% just based off of the nitric, right? So as it's transferring off to that, you're slowly going to get an even lower pH over those 24 hours, and then it comes back up again. Then you get that evolution of the nitrogen gas off the fact. So to answer Christian's question as well, and I don't know if the folks from Vital also do this or not, we actually make sure there's a very low head space gas gap at the top before we put a cap on. If we make a half-gallon bottle, I have like a centimeter of gas at the top of that bottle. So more than Pepsi would ship you a bottle of kind of situation. And it helps with that stability as well. There's not going to be as much of a gas exchange happening in that case.
And that's obviously what Christian is actually presenting. They accidentally found out, and we thought it was good to look into it. So
so I mean, but these additives that he's putting in after the fact are a great idea, and it's a way to take control of the water, of the water after the fact. So this is a great idea. I'm sorry to interrupt, to completely derail your story, but that
you're not at all. This is the whole purpose of this. It's making it. It's bringing scientists and people's perspectives in. You're absolutely not derailing anything, Ryan. We are so thankful for all your great comments here. This is wonderful, wonderful. All right, let me just continue on here if I can get my screen to work. We also did. Yeah. So we did another study where we, instead of quote unquote just adding 30 mil of 0.1 molar, we also looked at trying to change that so we get with 1 below and 2 above, and we did both sodium and potassium hydroxides here. And we again looked at the effect of time, the effect of base, and the effect of base amount. And if we look at nitrate, look at the red one, you can see that, as we saw before, we get a drop over time. But actually, if we add, say, 50 or 75 mil of sodium hydroxide, even though we have a little bit of activity there in the beginning, we can actually stabilize our nitrate concentration over time, which we find very, very useful. We see the same thing with potassium, maybe a little bit lower, but we are seeing the same thing. So we are now thinking about those being the target amounts that we should be going for. This needs more replication. This is only DI water. So we have developed a big part of this, of course, is science, is developing the methodology, getting the equipment right. So Miles has been doing a ton of troubleshooting, and now we got Bruno involved, which has helped us immensely. But Ryan, I think we need to have you quit your job and move a little bit further north and come and join us. So that would be a big step. But we are at least getting some insight into the dynamics and how we may be able to control some of that dynamic. So what you sort of alluded to and what we are getting into, and this is another brainchild of Miles, is the total nitrogen. So we have nitrate, we have nitrite, we have these different nitrogen forms. We need to have a good measurement of something reliable, reliable measurement of the total nitrogen, how it's shifting back and forth. We definitely want to also look at if we use these bases in the what we call the reduced PAW, how does that work on the crops? What is there a benefit to using potassium hydroxide, adding that potassium? Does that really give the plants a boost, which we are expecting? Maybe we could even get Kent involved in some of that, even though he's supposed to be retired. But I hope not. And of course, if we were to use some of these bases, what would that cost? So this is where Nate and Bruno are getting involved also, and where we need the companies to look at this. How would we inject this? How would we actually do that? Is this economically feasible? And maybe looking at other bases, right? So this is that we're just sort of scratching the surface here. So are there any questions? We've already had some beautiful discussion led by Ryan here. Are there any comments or suggestions before we move on to any of this stuff? Paul, are you at Vital Fluids? Are you looking into use of bases? Is this something you've considered?
Well, what we actually do if we use plasma activating water for fertilizing properties, we compose a complete tailor-made fertilizer recipe, which contains potassium, nitrogen, actually all the components that need to be in the water, because that is what the plant needs. So yeah, we add potassium hydroxide, I think it is. Yeah, potassium is also an important fertilizer for plants, but what we do is we look at the complete fertilizer recipe that is needed for plants to grow. We do that because there can also be an effect from other fertilizers on the pHW. So we have seen some precipitation that is unwanted. So there are, in that case, fertilizers are participating. So the recipe isn't good anymore. So we specifically make a recipe, a tailored recipe for a specific crop. That's the way we approach it.
Right. Okay. Any other comments before I move on here? Are we good? Okay. Good. So now I want to show some data that Patrice, my postdoc, has been working on, and I will be referring to two PAW, so you can see PAW 6 and PAW 9.4. That's the treatment time, and you can basically see the pHs of those two here and the conductivity as well. And we are referring to experiments where the irrigation, when we talk about supplemental PAW, they were supplied about 40% of the water was PAW. So that's quite a high amount. So this is probably not economically feasible, but still it's a start. And what we did here was we added either PAW to the bottom of to the pots themselves manually, or they got control water. Then Patrice meticulously collected leaf discs from these plants over time. And then he did what's called a life table study. And this is where I'm familiar, and where Ryan, maybe we can teach Ryan a little bit about entomology here, where we get into territory where I'm more familiar. So a life table study basically is bean counting of the different development stages. So you count how many offspring they have, how many eggs they lay, how long it takes for each individual life stage to be completed, and the mortality within each of these these development stages. So looking at it, what we really, if you think of time as our x-axis and population as our y-axis, you would expect something like spider mites or any other animals under unrestricted life conditions. You would expect an exponential decrease, and that's essentially what happens when spider mites infest a greenhouse full of tomato or marigold or whatnot, because food is unlimited and there are very few predators. What we are hoping to do, and the working hypothesis behind this study, was maybe we can slow that development down so that people can grow their crop before the spider mites go out of the spider mite populations get out of control. And or we can release natural enemies which will have a better chance of surviving and controlling the spider mites, if the spider mites are not running in 5th gear but only in 2nd or 3rd gear, if they develop much slower. So what did Patrice find out in his life table study? Well, there was not a big difference. There was not a significant effect of the immature spider mite development time when he looked at the 3 treatments. But he did see a highly significant difference in survival rate. So for some reason, when those spider mites were put on the leaf disc, they did not survive as well on the plasma treated, when the leaf discs were from plasma treated plants. So we had a reduction in survival rate of the immatures. We had a reduction in the longevity of the adult mites. So they live much shorter, considerably shorter. So you can imagine how many eggs a female can lay if she has 16 days to do it compared to 13 days. All the compounding effects of these parameters in the life table are really, really important. And also, if you look at the fecundity, so how many eggs they're laying, if you go from 50 down to 30, that's a big, big difference, okay? So if you take all of these variables that I just showed you, and then you run them through this program that Patrice knows how to run, you can actually predict how the populations will progress over time. And what you can see here is that the control populations basically reach their initial population after about 20 days. You can also see that was delayed just a little bit, but that the overall intrinsic rate of increase was much slower for the plasma treated plants. So if we look at, so the y-axis here is logarithmic. If you actually look at that, we can see what that, so that actually meant that using this model, Patrice could estimate that the population would be about 3,200 spider mites for the control, but it would be only about 250. So a much, much lower number. So guys, think of this as a pest management approach here, where you are lowering the pest population by 10-fold simply by irrigating your plants with plasma activated water. Now 42% of the water is what we provided. Maybe we need to go lower. Maybe we can do another PAW. This is where the story really starts getting interesting, right? He also did an actual validation where it was not leaf disc, where it was on full plants, and saw the same effect that on the, and this was replicated. So you can see that the so the model was actually corroborated by whole plant validation, which is rather extraordinary. And just to this is more of academic interest, but really hones in the message here is there's this ecological hypothesis called preference performance hypothesis, or in modern terms, it's called mother knows best, which is essentially that a female should prefer to lay her eggs on the host on which her offspring will have the highest performance. So basically, if she can assess the difference between two options or three options, she should lay, natural selection should favor her laying her eggs on the host that is most suitable for her offspring. So what Patrice did was he took those leaf discs, did them in pairwise comparisons, the control versus the PAW. And he showed, just like we would expect, that the control were, you can see that in the first comparison here, they were significantly more they were settling more on the control. They were also settling more on the control than when they were given the other PAW. But when they got the choice between the two PAWs, it was sort of equal. So there's clearly a difference in settling. He also did it for eggs and showed the same trend. So she would prefer to be there. She would also prefer to lay her eggs on the control over the PAW. So that obviously raises the question here: what is going on? And well, actually, before we get to do that, let's just show some other results that he's working on. So this was on tomato, but also on marigold, we are seeing as somewhat of a difference here, that they prefer the control over the PAW. On beans, there was also a difference at 2 time points. What is interesting is that on strawberries, he did not see a difference in the preference of the spider mites. So if we just to summarize here, if we talk about morphometric characteristics, I showed you that earlier. We have clearly, and we also look at reduction in spider mites. We can see that we actually found that in terms of morphometrics, so this is the size of the plants, we did not see a PAW effect on soybeans. In terms of reduction of spider mites, we did not see a reduction in strawberry plants. And we actually think we can explain this because soybean is, of course, a legume, so it's nitrogen fixating. So PAW may not be such a big contributor based on the fertilization regime we were providing. Maybe the soybean plants are not benefiting from this. So this, for you guys who are in the commercial business, maybe PAW is not so much of a you shouldn't be targeting legumes. But when it comes to spider mites, we also think we can explain why strawberry plants were not, we didn't see a PAW effect there. And we think that has to do with strawberry plants having significant amounts of trichomes already. So maybe there is something there that suggests what is the effect. Because if we look into the mechanistic effect, what Patrice showed on the tomato plants was that the trichome density was significantly increased by the PAW applications. So this is why, Ryan, you need to go back to your cannabis growers and say you can get stronger weed, you can get more, you can get better product by potentially using PAW, not just the amount, but also the quality. So this is something we really think is important. What Patrice also did was he did element composition of the different plants. On the tomato, what he saw was that there was a significant increase in the leaf concentration of nitrogen, potassium, phosphorus, calcium, and magnesium. And there was a significant decrease in zinc and iron as a function of this PAW irrigation. So we are really sort of getting an idea of some of the mechanisms that are behind this. we are now with some of the undergrads in my lab. There's one student, Sammy. I should mention that this has been published very recently. So you're welcome. If you're interested, you can contact either Patrice or myself, and we can send that paper to you. What Sammy is very interested in, she's very interested in cell biology and histology. We're actually going to start chopping spider mites into fine slices and in wax, and looking at the organ development in the spider mites, because we have reasons to believe that one of the reasons why this. So if we saw a lower fecundity, well, we want to show that histologically. We want to demonstrate that maybe the ovaries are underdeveloped in spider mites that have fed on PAW treated plants. So this is just getting into the nitty-gritty part of how spider mites are affected by the PAW. So we're getting this life table work and is very, very meticulous, and Patrice is an absolute maniac in the lab and has been spending, you have to check them every day. You cannot miss a single time point. So this is extraordinary work that he's doing, and he's supervising some of these students getting involved in this. So this is, we're really getting ahead of the curve here and providing some insight that I think is very important. And for you as companies, for you to support your marketing strategy, get some that this is not snake oil, that there's some meaning behind this madness, that there's getting some quantitative data on this, I think is very, very important. So we are one of the frontiers we're looking into is the performance of PAW, how it affects predatory mites. With the quality of these mites that are growing on PAW plants, are they as suitable? We want to look into that. So we're working with some of the commercial producers of predatory of beneficial insects. So Coppert Paul is one of the so that's the Dutch company that we are definitely working with. We are also looking at PAW, integrating it with light regimes. So this is where we are taking the PAW and thinking of manipulating, spraying it onto plants, but actually integrating it with use of LEDs that manipulate the pests up into the canopy where they are more susceptible. So I'll show you some data about that here in a little bit. But at this point in time, are there any comments to this presentation, which is squarely based on the great work by Patrice in my lab? Any comments to this?
Yeah, I just had a comment. So on the slide where you had the mineral content in the plant, I think that you had a question about the mechanism. So when it comes to mineral content, its use or uptake by plant or humans is a lot dependent on the pH. So right. For instance, ion 2 is the form in which we need it. Ion 3 we cannot use. And so ion has to be in an acidic environment in your stomach. So I think a lot of this is being kind of controlled by pH. Yeah.
Right. Absolutely. So Patrice did this great work with 2 PAWs, right? But there's no reason, once we start adding bases and different, I mean, there's so many layers to this, right? We will continue working on this, but we definitely have a methodological platform now to produce lots of articles and lots of knowledge about the PAW and how it impacts not just spider mites, but also other key pests in greenhouse production systems and elsewhere. Yeah. Any other comments? Thank you, George.
Yeah, this is
all right.
I have a question. Have you looked at this? Yeah, have you looked at this on thrips? This is interesting, but we have.
So we will get into this in a little bit. We are looking at thrips, but I have some data on thrips, but not on the indirect effect. I'm sure that if you teased Patrice enough, he would jump straight on this and get even less sleep than he's already getting. So we will be absolutely. Right. Thrips are on our radar, and as you will see here in a second. So what I want to go to is this is me to the left. I'm a certified sprayer, and we do a lot of industry trials like this, where we spray pesticides. So we have a lot of knowledge, have a lot of industry collaboration in this field. But thanks to Anil, we have also developed a commercial spray rig like this, where we can modify nozzles. We can go at different speeds. We can go at different heights. We can go at different boom settings and different pressure. We can simulate any ground rig application. So similar to what you had there in your greenhouse, Paul, we have those experimental settings. And there's no reason why we cannot spray PAW either by itself or in a mixture with pesticides. So that's definitely something we're going to be, what we're going to be looking into. And we already have some data, again, with Patrice as the main workhorse behind this, where we use pesticide as a carrier. So we are thinking, Tom, to address your question partially, is why use water when you can do PAW? Why? Maybe Paul and Ryan, maybe for your companies, there's a market for you to sell water to be used as a pesticide for pesticide formulations. Okay, so instead of using regular water. So basically, what we showed here. So this is spider mite mortality, where we mixed it with neem oil. So you can see if we use the water by itself or we use deionized water, we get hardly any mortality, but we can get decent mortality, especially if we. It seems like there's a good reason for adding neem oil. So in cannabis or in these other crops where you have restricted amount of pesticides, or if you are an organic user, use these organically certified pesticides that are not very potent, not very, very strong. Well, add a kick to them. That's what we are really hoping to demonstrate. And we are getting data that support that claim. So we can reduce both number of eggs and also, and here are your thrips, and here's your marigold, okay? So what we're actually seeing is something kind of funky, where the PAW is performing very, very well by itself, okay? Maybe even better than it does when we add neem oil to it. We're not quite sure what's going on here, but we are definitely going this route, Tom. So we are very interested in doing this work, and we are collecting data on it. We of course need to look at the phytotoxicity. What Paul shared with us seems to suggest that there's probably not a lot of that, but let's see. We will continue working on that. We want to see how and take it to yields. We want to do more commercially relevant trials. Look at the fungicidal effects, which I know that Vital Fluids is very, very interested in. So that's certainly also something we want to pick up on. So are there any questions to this? This is just kind of short and sweet, and I think we're doing great on time. If not, let me just continue. I have a few extra slides, and then we can sort of have a powwow towards the end here. I want to go to lighting, because I think it's such an incredible opportunity, with more and more of our food grown inside, with climate change making growing conditions more unpredictable, and with more and more food being grown in urban areas. We need to look into that, and applying PAW in a contained environment, of course, is much, much easier when you have a structure. And it can then be combined with these other conditions, such as lighting. And so we are working in my lab, and I know that Neil is also working with some of that. We using different light regimes to manipulate pests in different ways. So can we pull them from the bottom? So spider mites are very, very abundant, very prevalent on the abaxial side, so the underside of leaves in the bottom of canopies, and they cannot be targeted with systemic insecticides. So you have to spray something from the top, but the insects or the mites are in the bottom. I'm sitting here, my hands are going all over, but I'm not even showing my video here. So maybe I'll just see if I can get it going again. So what we are trying to do is, can we use light in some way to manipulate them upwards into the canopy? This is something we're very interested in, and then hit them with an organically certified pesticide that is in which we use PAW as a carrier, right? That's kind of our goal here. So we have done some work on Western flower thrips. If we subject those leaves to either from the top or from the bottom or a day at night, it doesn't really matter what time, we can significantly reduce the hatching rate of Western flower thrips, and maybe some of this can also have some fungicidal effects, because we think some of these doses of light can actually be lethal to fungal spores. Maybe not near infrared, maybe in the other end, maybe more towards the UV light. So we are also working with that. And one of the things we have done is, under experimental conditions, followed how mites move around. So this is another Dutch invention. This is etho vision. So Paul, your country is all over this. Very impressive. So you have this video system that can monitor them, and we can expose them to different light regimes. You can see over here that these bars hold different light regimes, and we can move them around so we can see how much they move. And if we have the mites moving around without a shelter, we can actually, there's some statistical analysis that still needs to be done. But overall, we are predicting that mites that would move more if they're trying to get away from something that is very unattractive to them. And there's a little bit of a trend where they seem to be moving more when we give them certain light regimes. And so we can do that in that arena without a shelter. We can also do it when we have them inside a shelter where they can move in and out. And we actually see there that the mites, interestingly enough, they sometimes stay in the shelter and sometimes they shy away from the shelter. So what we found out here was that the blue light, they really like to stay outside the shelter. So they are attracted to blue light. And we also saw that they were, at least they did not prefer the UVC, which of course is also what you would expect. We protect ourselves from UVC, so obviously they do the same thing, go under the shelter. Now we don't want to use UVC because we can kill the plants. That's plants don't like UVC. So we set up a separate experiment where we used blue light and UVB so we basically take UVB from the top and then UVB from the bottom. So we repel them, and we attract, and we pull and push, move them upwards. And if we do that for 10 minutes prior to spraying a Botanigard, which is an organically certified pesticide or miticide, we actually, and do that on bean plants, we can actually go from we can get a very, very strong effect so we can actually dramatically increase the effect of our organically certified pesticides. So we think that is really, really interesting. So we want to look at lights. We want to look at how it can affect other pests, the benefits, the economics of this. So that basically summarizes the discussion that we have prepared here, and we are about half an hour before conclusion. But I think we're all I think this has been enormously productive. I think it's been a fantastic discussion. Are there anything that people would like to bring up at this point? Are there any comments that people want to make?
I would say this is great, especially with the feedback we're getting from members of this meeting. Ken, of course, I'm digging into the comment that you actually made, but Christian, this is great. I think we should continue to do this. I really have enjoyed the depth of the presentations and also the discussions. This, as I said, should continue. Maybe, I don't want to say more often, but kind of regularly.
Let's regroup tomorrow. Let's continue tomorrow. Yeah.
So we kind of update ourselves with what the industry is doing.
Right.
I particularly like the fact that you had industry present, so we know what is also going on. And that really helps us to think about what kind of research we should do. So great, good job.
We don't want to we don't want to tire people, so we don't want to waste people's time. So we will do this when we think we are not going to set a date, but I can assure you that we will continue having these meetings. And as I said, we have just Anil, give us an update on the tomato field, the tomato plants.
Yeah, so we transplanted, I want to say about a month ago into the field, and now the plants are maybe 12 to 16 inches. And we are definitely seeing a difference between the treatments. Something that we have done, which is best for science, but I don't have the treatment numbers memorized, so I cannot confidently say which PAW or what treatment is doing better than the other. But just based on the fact of everything, all the other knowledge, I can pretty confidently say that some PAWs are doing much better than the control.
So we have field. So what you're saying, and just to reiterate, we're not using PAW in the field because we think that is economically that's not feasible. We are strictly talking about the transplants were treated with PAW, and then we stop. Then they are all getting the same fertilizer, the same irrigation. But you're saying just from visual inspection, you're flying a hyperspectral drone and an RGB drone over this. We're doing all sorts of measurements out there. We're going to get data, yield data sent to George and whatnot. But you're saying right now it looks like and how many PAW? I think there's 3 or 4 PAW treatments out there, right?
I believe there's four treatments with and without the different types of bases. So we have one,
and this is one soil. This is one soil type. It is one fertilization regime. But this is where, with Tom's expertise and contacts and whatnot, we will do much. We will go down that route, and we will. This team will get there, and also with Neil and Nate and other people working in greenhouses and whatnot, we will get data not only from California, from elsewhere that have commercial relevance for sure. With that. Any final comments by anybody before we adjourn here?
Good job. Final comments.